It's something that takes a little balancing. It takes a little getting the right amount so that you're stopping the thing from damaging the pancreas while still allowing some digestion to go on. Yes, sir?
Is it all or nothing, or is it more sporadic, and how many of them are active?
Good question. Is it all or nothing? What happens is once this guy is active, it can start attacking inactive ones as well. So it tends to happen fairly quickly. And it tends to be pretty much an all or none. You don't see different levels of that happening. Yeah.
So once it's treated, though, you're always going to have those active ones in your
No. Once it's been treated, you can be cured of it. So it's not like you're always going to have that there. Okay. But what you have to do is get the sort of system cleaned out of what was there. You may be prone to having it again because maybe you had a duct that, for example, let things leak a little bit. And so once you cure it once, it may come back again. But it can be cured, yes. Okay, so let me give you a little terminology. So I said a zymogen is an inactive enzyme. A zymogen becomes an active enzyme by peptide bond breakage. It takes breaking of peptide bonds to convert a zymogen into an active form. Okay? We tend, and it's not an absolute rule, but we tend to refer to the zymogens of a protein with the letters OGEN at the end. Okay? Chymotrypsinogen is the zymogen form of chymotrypsin. So the OGEN indicating that. Not all zymogens have that, unfortunately. Okay? We're not going to pick apart and get which ones don't and so forth. But in the modern nomenclature, the OGEN is used to indicate the fact that it's the inactive form of an enzyme. So chymotrypsinogen is the inactive form of chymotrypsin. Trypsinogen is the inactive form of trypsin, etc. Okay? Yes? Yes? It is. So the question is, does another enzyme create that? And chymotrypsinogen is an interesting one. It turns out that it has to be cleaved first by trypsin. Trypsin is an enzyme that we have present abundantly in our digestive system. So once this guy gets down to the digestive system, it becomes activated. And you say, well, is trypsin, how about it? Is it made in a zymogen form? Yep. So trypsin, when it's made, trypsin is made as trypsinogen. By the time it makes it down to the digestive system, again, the protease acts on it. So you start saying, well, what's the first one, right? How far back do I have to go before I find the inactive one? And there's a peptidase called enteropeptidase that appears to be the master peptidase or master protease that governs this whole process. But it's beyond where we're going to talk about here. Enterokinase, no. Enteropeptidase is the name of it. A kinase is something that puts a phosphate on. Yeah. Okay. So let's see. What else I want to say? Oh, as I started to say, that chymotrypsinogen is interesting, and it's different. It's activated first by trypsin, but you can see it's a two-step process. So the first step involves cleaving of a single peptide bond right there, and then that makes a form of chymotrypsin called pi-chymotrypsin. Pi-chymotrypsin is partly active, and the only thing it works on is more pi-chymotrypsin. So at some level, once you light the fuse, it starts I shouldn't say self, but one pi-chymotrypsin will work on another one, etc., to activate them into a chymotrypsin form. So chymotrypsin is an unusual one in that respect. Okay? Now, I'll tell you another system. Your book doesn't go into it here. I want to tell you about another system that uses zymogens that you need to be aware of that I think is fascinating. Okay? And this form is what you have in your bloodstream. Blood clotting happens as a result of protease action. Okay? Protease action. You have in your bloodstream clotting factors that are not active. It's very important that they not be active, because if they are active, they start to form a clot, and you certainly don't want a clot in your brain or in your heart or something like that. So you want to have these things floating around and inactive and ready to form a clot when you need it, but you don't want it forming a clot when you're sitting around like you are here. There are people who are much more prone to having clots. They're much more prone to having that activated. They may be very prone to stroke and or heart attack as a consequence of that. So zymogens are present in our bloodstream as clotting factors. The wounding of the skin causes a series of events to happen that activates those zymogens. And when it does, the clot forms from the cleaved proteins. The clot actually happens as a result of polymerization. And it makes a mesh. It's a really phenomenal process. We don't have time to go into it here, but it makes a mesh once a little piece of the precursor protein is clipped off. It can't make this polymer until this little piece is clipped off. But when it gets clipped off, now it can make a polymer, and that polymer makes a fiber that holds back the blood and keeps blood from coming through, and that's what makes the clot. It's a protein called fibrin. And the inactive form of it is called fibrinogen. So fibrinogen is the inactive form. Fibrin is the form that makes the polymer. Okay. Makes sense? Yes? Yeah, I haven't talked about it, but there are a series of proteases. It turns out that in the blood clotting system, there's a series of about 10 proteases that do what's called a cascade. One activates the next, activates the next, activates the next, activates the next. They're called factors. So you got factor 7, factor 8, factor 7a, factor 11, et cetera, factor 9, that are proteases, one activating the next, activating the next, activating the next. And you don't need to know this. I'm just telling because he asked the question. And the last one is a protease called thrombin. Everybody heard of thrombin before? So prothrombin is the zymogen form of thrombin. That's where they don't use the O-gen. So prothrombin is inactive. It becomes activated to thrombin. Thrombin cleaves the fibrinogen to make fibrin, and the clot forms. I'm sorry?
Is vitamin K in there somewhere?
Vitamin K is in there. Yeah, yeah.
What are platelets for?
I'm sorry?
Where's the platelets for? Is this containing those sometimes?
Platelets are blood cells that facilitate the clotting process because they are relatively sticky to each other. So there's a fairly complicated process in forming the clot, and the platelets do help to play a role in that process. But this is a molecular process I'm talking about here. This is a protein that's actually it's present floating around in the serum of the blood. Okay, let's see. So that's what I wanted to say. Now I'm going to turn our attention a little bit to thinking about how enzymes do what they do. So we talked about control mechanisms. We talked about how they're turned on, how they're turned off. Now we're going to spend a little bit of time talking about how they catalyze a reaction. And I will tell you, I'm not a fan of mechanisms any more than you are. So I'm going to try to keep this as simple as we can, but we still have to talk a little bit about mechanisms so we see a little bit about how enzymes actually lower that transition state energy. Okay? All right. Well, let's think about chymotrypsin. It's one of the most studied enzymes around. And it's studied because, as I told a student in my office earlier today, because biochemists are lazy people. Okay? It turns out that chymotrypsin can work on a molecule that is man-made such that when chymotrypsin acts on it, a yellow color is produced. Okay? A yellow color is produced. That means, then, that by measuring the amount of yellow color and how fast it occurs, we can follow the reaction that chymotrypsin is catalyzing. So it's very simple to do. We have machines called spectrophotometers that can monitor the production of color quite readily. We put chymotrypsin in there with this molecule, and we say, hey, what happens? Well, what we see happens is interesting. Okay? So the molecule's name doesn't really matter for our purpose. All we have to worry about is that before chymotrypsin works on it, it's not yellow, and after chymotrypsin works on it, it is yellow. When we look at the production of yellow, we see something interesting. We see that yellow is produced very rapidly, and then we see it slows down. So we see a fast rate and a slow rate of production of yellow. Now, when people first discovered that, they didn't quite know what that meant. Okay? Didn't quite know. Yes? So the substrate that the chymotrypsin is acting on. So thank you for asking. The substrate is colorless, but after chymotrypsin acts on it, it becomes yellow. So the product of the reaction is yellow. So how do we explain the fact that this reaction went fast and then it went slow? Well, people thought about it a lot and came to the realization that the reaction was happening in two steps. A fast step and a slow step. Not surprisingly. Okay? So we see chymotrypsin works by two steps. One step that occurs very rapidly and one step that takes a longer period of time to go. Well, let's look at what that means. Okay? Here's the reaction that this guy's catalyzing. We don't need to worry about the structures. This is the yellow product that's right here. Okay? This is what is being produced. Okay? Oh, back up. All right? And what we find in looking at this is here's the intact molecule here. Okay? So this is the starting substrate that has no color. Here's the enzyme. It hasn't bound it yet. We discover that when the enzyme binds it, very, very quickly, the enzyme breaks the bond between this guy right here, okay, over to here, and this. So the enzyme becomes covalently linked to this little piece, and it releases this piece. This step happens very, very fast. And this is producing yellow color very, very fast. Well, remember that a catalyst is something that that catalyzes a reaction but doesn't get changed in the reaction, right? I've already told you that enzymes get transiently changed. That was the induced fit model, right? It transiently had its shape changed, and that helped it to catalyze a reaction. Here's a definite change. Here's an enzyme with nothing bound to it, and here's an enzyme with a covalent bond to a part of the substrate. This guy is not back in its original state. So the second part of this process requires that the enzyme be returned to its original state, and that requires release of this material right here. That's the slow step. So the fast step involves the enzyme becoming covalently attached to this with the release of the yellow color. And then the second step, this covalent bond has to be broken by water to release the acetate. Then we're back at the enzyme, the free enzyme, and we're back where we started. Yes, sir?
So if there wasn't water present, would the second step not happen?
It would not happen. However, remember, enzymes are dissolved in water. So water has to be there. Yeah. Good question. Make sense? Slow step, fast step. So the fast step involves making a bond between the enzyme and the substrate. The slow step involves releasing that bond. So the enzyme can go back and start the whole process all over. Okay. Yeah, that's a good question. It actually happens fairly quickly. Okay, this is a relative time scale, but this would happen in the order of seconds. It doesn't take very long. So you have to be looking at these things happening very, very quick. Do your measurements very, very quick, or you would never even notice this part right here. All you would see is just things kind of going up like that. Yes, sir. Well, good question. Does this ever flatten out or keep decreasing? As long as you had sufficient substrate, it would continue going up because all we're measuring here is time. But eventually, in a typical reaction, you have a fixed amount of substrate in a tube, so it actually would level off over time. Yeah? This is the yellow making stuff. That's the fast step. And then what? So the dotted line is just showing the release of that molecule from the enzyme. So that little 2-carbon piece, that's this. I didn't talk about it, but that's what this is here. So they're showing you the release of this, and it parallels what's happening here. Okay? So during this period right here, the enzyme has got to get itself free of that acetate. If it doesn't get itself free of the acetate, then everything slows down and stops. Okay. Now,